Thanks so much, Dr. Good. And with that, we'll also slide in right into our panel discussion. So I asked Dr. Shapiro to join us as well, and he is awesome. We definitely got a lot of questions, but you two being kind of the focal speakers in that session, I'll also give you the chance to maybe ask a question as well if you have one before I'll start asking. Okay, otherwise I will just start with a few questions from the audience. So I think the first question we saw is from Tammy Lieberman. It's directed at Jesse Shapiro, and she says, "Fun to see reanalysis of our data. Can you please clarify how you calculate gene loss in the B. vulgaris time series? How have you separated loss from variability and ability to detect genes across time points?"
Yeah, can you hear me? Yeah?
Yes, we can.
Great. Okay, so yeah, so this was done using MIDAS, okay? And for clarity as well, this is all using metagenomic data, not the isolate sequences. And yeah, so this was okay. Yeah, so I'm just looking at my slides. Hang on. Guess I can go back to the slide. Okay, right. Can I share my screen again just quickly? Maybe this will be, okay, yeah, okay, good. And look, okay, so you got, so, okay, right. So we're defining copy number, right, which I didn't define, so thanks for the question, right? So this is defined, and you can see this below in the speaker notes here. So we have the depth of coverage of a focal gene normalized by the median depth across 15 universal single copy marker genes in MIDAS. So that's what gives us this concept of copy number. Is it's kind of like a deviation from the median coverage. So when it goes close to 0, and there's a bit of wiggle room here, so it's less than 0.05, that's our definition of absent. And then when it's around 1, then it's present, and it actually can't exceed. It can't go too much over 1. There's some other filters as well where we blacklist genes that are too far above the median, both for the SNV changes as well. So basically, we try to get rid of genes that are changing their copy number too much, so they're actually in duplicate or triplicate. And there's also a blacklist. So this is following actually from Ben and Nandita's PLOS biology paper, all these filters where any reads that map to other species as well, we throw out, so to get rid of sort of mismapping. So there's a whole bunch of filters that go into this. Yeah, I think that these are pretty reasonable filters. There's always the chance that there's some noise in here, but that's basically what we did. I hope that's clear. Thanks for the question.
Can I ask you actually a follow-up question to that, if you don't mind?
Sure.
So I guess with 2 time points, gain and loss sort of makes sense to me. But then when you have 50 in a row, do you try to assign like a single call for every event? Like if you get lost, that means you can never come back up later, or you sort of, for every pair of time points, you're recalling gains and losses every time? Yeah,
that's good. Yeah, so you are recalling, which means, so yeah, if it goes away, it can come back later. So it's always just between a pair of time points. And yeah. So I guess you can definitely, the loss, it's certainly like a limit of detection, right? So maybe it is there, but too low to call.
Okay, perfect, thanks. Yeah, our next question is directed at Dr. Good. It's from Varun, and they asked, how do we define reference for a species? Strains have massive diversity, and do such mapped as SNPs even capture real dynamics? And yeah, he added as a clarification, like, how do you also define strain in your approach?
Mm-hmm. Yeah, it's a great question. So I guess the way I think about it, the analogy I always try to tell people, it's like a little bit like Plato's cave. So, you know, when you're using reference-based approaches, already from the very beginning, like you're only looking at half the genome, right? Because strains differ so much in their gene content, you don't reliably capture all the genome that's actually there in your sample. And so a lot of what I talked about today focuses in even more particularly on the core genome. So we're only looking at really boring SNPs on the core genome. And so the analogy I like to give is sort of Plato's cave, where you're really just seeing the shadows of all the actual like interesting functional variation that's happening. So we don't know in any of the stuff I talked about today, like that those are the causative changes. In fact, some of the work we've done suggests that there's so much genetic linkage in these microbial populations that it's really difficult to distinguish what the true, you know, causal mutation is for any of these functions. But that almost helps us in our sense, because that means we can look at trajectories, even of really boring synonymous mutations going up and down, and trying to learn something about, you know, the actual selective differences that are being selected, maybe on a gene gain or loss event that we just can't look at in the reference genome. Strain for me, to get to the second part, I guess, it's a little bit of a complicated question. Folks have written lots about this. I guess I try not to define it so much in terms of is it a strain or is it not a strain, but to think about the time scale over which two different clones are related to each other. And I do think you can say something interesting about if the time scale is much longer than a human lifetime, you know that had to come from outside the host. And so it's a different kind of event than if it could have accumulated within a host over a single host lifetime. I think it's really hard to distinguish between a strain or maybe you'd call it a clone or something within a single host, because there's just a lot going on there.
Okay, thanks. Yeah. The next question is for Dr. Shapiro again. It's from Anonymous, and they ask, time lag, decay, and SNV turnover or gene loss is interesting. Is this more consistent with correlated events, e.g. strain turnover rather than de novo events?
Okay, that's a great question. Yeah, so we do see this decay, especially in the relationship between community diversity and changes in SNVs. So we see it more between, you know, close time points up to about 50 days, and then it drops off. Is that, yeah, so I don't know if that speaks directly to strain turnover versus de novo. You know, my feeling is that just based on like the axis of how many SNV changes we're seeing, it's in the thousands. So I mean, it's real, like most of that is strains. So I think that's the main line of evidence, combined with other work, and actually some of this is in a preprint from Ricky Wolf and Nandita Garud, also reanalyzing this data set, as well as another paper from the Alm lab, their microbe-seq approach, which shows that there's probably about 3 fairly abundant strains of B. vulgatus. So there's some evidence that there is strain diversity. So that's kind of the line of evidence of, that's what we're looking at. Yeah, but I don't know, in terms of like that sort of decay, if you sort of expect to see that more for strains versus de novo, I think, so to go out on a limb a little bit, right? So I think our sort of conceptual model here is that, there are these strains that are there. And this is, again, n equals 1, right? This is one individual. There's another individual that we looked at. I didn't show you those results, but it's fairly similar. So there are these 3 strains. They're persisting, right? So none is driving the others to extinction, but there's these kind of subtle fluctuations in which is more abundant than the other, and that that tends to correlate with community diversity. Okay, so that's the observation. So I think, given that those strains are there, it's I guess it can be a pretty quick response for one of those strains to kind of go up, and Ben showed that as well with the antibiotic perturbation. You can just, like, within a few days, you can really see a massive shift. I don't think we're seeing anything so massive, but just, it can happen quickly. So in that sense, I think that the strain abundances can shift pretty quickly. How does that relate to a de novo mutation? If it's really a de novo mutation to go from appearance to, an appreciable frequency, maybe that's slower. And I guess you get into sort of the population genetics of, maybe it appears, but it doesn't, it doesn't pass the drift barrier. So it takes like a few tries to get it. So anyway, that's a bit of speculation. I don't have the real answer to that, but those are some thoughts.
Okay. Yeah, thanks. So I'll ask one more question specific to Dr. Good, and then we will follow up with a question for both speakers. So Dr. Good, that question is from Emmett, and they ask, do you know which bacterial strains are accumulating these intense amounts of mutations? So, and whether that's specific to particular taxa or is kind of ubiquitous for strains in the microbiome?
Yeah, it's a great question. Here I have a backup slide actually for just that question. Let's see, can I share? Yeah, so we looked into that a little bit, and here you can see a little bit of the data. So what I'm showing you here are I guess different microbial families and the probability of a genetic change. This is both an evolutionary modification or a strain replacement event, right? Sort of two different biological things going on. And so, the problem, of course, there's a lot of different species. We're able to get such good power in the first place by aggregating across the different species. So in this, first data set, there actually isn't a ton of signal for many of the species, but actually those that are a little bit more abundant, you can see some systematic changes. So you see, and the colors here, the blue are the rates of evolutionary modifications and the red are strain replacements. And so you can see actually there's a pretty big difference between the Bacteroidetes and the Firmicutes right off the bat, that actually in both cases, they're a little more likely to acquire a strain replacement event, a little more likely to acquire an evolutionary modification event. The specific species involved, that we just don't quite have the power to look at that yet. But at these really coarse levels, there are some interesting trends there.
Awesome, thanks. Yeah, and so for the next question, I will paraphrase a question that kind of in different forms appeared a few times in the Q&A, and it's basically related to the environmental selection pressure that might be associated here, and that will be directed at both speakers. So for instance, do you see any associations with rapidly changing environments or environments that seem to be more stable over time? And also, especially for the gut microbiome, for instance, an association with dietary patterns or the diversity of carbon or nitrogen sources that are consumed by the individual.
Yeah, I can comment on that first, which is first actually to thank Ben for that great slide, about direction of causality, right? So how you can have abiotic factors that then are driving, both evolution and ecology, right? And so it's really hard in these observational systems to distinguish that. To some extent we can measure changes in diet or antibiotics and things like that, but it's very hard to get at those sort of causes and effects. It's certainly a really interesting idea to try to look at environments that have some kind of measure of environmental change to see if that would have an effect. It's hard to, I guess, short of modeling it, and maybe Ben has comments on this, but it's hard to get that level of data from real observational studies, I would say. Or if you have things in mind, I'd love to hear about it. But yeah.
Yeah, I would largely agree with that. I find both the modeling and the laboratory approach really important here, because those are the one case where you can actually control what's happening in the environment. And I don't know, at least to me, I guess in these larger communities, this distinction between external versus internal gets a little weird too. So actually the one example I talked about it in my talk, this one where we saw the selection pressure happen within 2 days, right? Now, on one hand, you might say, well, that's because there was this antibiotic perturbation. But actually, if you look at that one, we were really lucky to have really good dense sampling right after antibiotics, because that one actually didn't happen during antibiotics at all. It actually happened like 2 or 3 days after treatment had completed. And so what's happening there, our sort of theory for what's going on, is that, well, antibiotics cause this really big shift in species abundances. That changes the sort of local environment for the focal species. And that itself was sufficient to drive really huge changes in relative abundance on a really short time scale. And so I think once you start thinking of the environment as both the external thing and the other species, this distinction between environmental versus internal, it's a little hard to pin down, but very fun to think about, I think. Yeah. I guess, sorry, one more quick thing to add on to that, sort of going back to the question Jesse was answering before. I think what's also really instructive for me is to sort of try to write down the timescales over which you expect things to happen. And so actually the calculation that Jesse suggested of, let's say you switch diets today, and then you stick on that diet for a week and switch back to your old one. How long do you think it's going to take for a new mutation to appear and take over? Depending on how big you think these gut populations are. If you plug in the census population size, the time it takes for a new mutation to get up to something you could actually see ends up being on the order of like several weeks, even for a 10% fitness benefit per day, which is pretty big. So I actually think a lot of these environmental fluctuations may be a little less severe than we might think, just because the community is so big. Worth thinking about.
Yeah, thank you, and sorry for the interruption. And yeah, maybe to add in another question for both speakers, there were also a few questions that were not related to the environment, but more like to where those particular variations occur in the genome. So did you, for instance, look if that's like kind of homogeneously distributed across genomes, or if that diversification processes, for instance, in Dr. Shapiro's talk, affect like particular regions of the genome or particular genes, or is it kind of like a more generalist process? And, do you think it can actually even be mapped to genes like that, or is it definitely hard to do?
Yeah, that's a great, the short answer is we haven't looked at it yet. We like similarly to Ben, we're mainly for SNVs, we're looking at synonymous diversity as well. So kind of like the initial goal is not to find the targets of selection, but really to just find the shadows, like the evidence for those changes. Certainly, in these case studies where, we do see these gene changes in B. vulgaris. I have the list of those genes of like what are the genes that are being lost, or that certain strains have and others don't, and that are kind of responding. At that level, I think it's hard when it is these strain effects. I think it can be hard to find the targets of selection, because it's kind of a lot of genes. So I actually haven't looked into that in detail, but it'd be an interesting thing to try to do. But yeah, given the fact that it's a lot of species, and cover the coverage might not be even along the genome, so you might not see the targets of selection. Still, no, I mean, yeah, for sure. You could try to look for parallelism, like if there are genes that are targeted across different species, for example. Yeah, but the short answer is we haven't done that yet, but it's an interesting thought.
Yeah, I guess I'd mainly echo Jesse here, and maybe make a plug for Jay and Tammy's really awesome paper. I think like the experimental design you want to have, if you really want to look at sort of functions, you just need a lot of replicates. You can look at parallelism. And so for folks who aren't familiar with that work, they did isolate sequencing of a large number of B. fragilis clones. So the trade-off there is you get to look really in depth at one species, but you do then get to start asking these questions about like, are similar things selected across hosts? So they found some really cool signatures of, certain polysaccharide utilization loci, I think, getting targeted, many times independently in, the same hosts and different hosts. I think maybe the interesting thing there to me, if I remember correctly from looking back at their data, many of their signatures of positive selection weren't necessarily associated with mutations that took over the population, whereas like the stuff Jesse and I are talking about today, these are mutations that managed to completely fix. I think that raises some interesting questions about, there could be a lot of stuff sort of under the hood here that we're really not seeing in these sort of population-wide metagenomic kinds of approaches.
Yeah, and just for context there, right? So we focused on B. vulgaris mainly because it's just well covered. Like it has over 20x coverage in all, in these hundreds of samples from a single individual over time, right? So even at that level of coverage, if that's like the mean coverage, it means that there are, in certain time points, there are certain loci that are dropping out, just because they're not covered. So I think even in that, it's hard to find the targets of selection, and that's just in one species, right? Where for others, B. studless, in the metagenome, you don't have enough coverage to do that kind of analysis. You need to do the isolate sequencing. So I think it's very like broad broad snapshot, but hard to needle down into the specific adaptations.
Yeah, I mean, it makes you wonder, maybe an interesting approach then is to, like, if you have some hypotheses for about like what things could be targeted, to do like super deep amplicon sequencing just of those genes. That would then help you get more species involved and maybe see, like, is that pattern B fragilis or, you know, general across all Bacteroides or not. I think that could be an interesting approach there.
Yeah, for sure.
Yep, great points. Maybe we'll go through a few more questions from the Q&A. We have one that is specific for Dr. Shapiro, and it's from Anonymous. And I didn't ask that question, even though I'm probably to blame for it, because it is, have you thought about metabolic modeling as a means to interrogate the mechanisms driving DBD? That, I think, is in reference to the course yesterday, since we looked at how to predict ecological niches based on the genome sequence.
Right. So the answer is no, but other people have. So I guess, like, you know, maybe Ben wants to respond to that too. I don't know if you would call, he would call his model a metabolic model. But there is a paper that just came out in September from Andreas Wagner. So Magdalena San Roman and Andreas Wagner in molecular ecology, which does use a specifically metabolic model. Here, I'm just looking at it. I'll show you just quickly. So the pitch for this paper. So yeah, there it is. Which is really interesting, and actually finds with these models very similar results of sort of a DBD, like, you know, positive feedback, accumulation of diversity early on in community assembly, but then eventually reaching a plateau where you just reach a ceiling of the number of niches that are available. So I haven't done any metabolic modeling. But others have, and it seems to give pretty concordant results. So that could, you know, I think in terms of mechanistic explanations for this, I think it's sort of plausible and worth pursuing.
Yeah, awesome. And as a related question, and that one is directed to Dr. Good, Sean also asked, can you discuss how a resource model might relate to Dr. Shapiro's DBD work?
Yeah, that's an interesting question. Let me pull up my slide here at the end. Sorry, I need to share the screen, don't I? Okay, right. So you know, we're using this relatively simple resource competition model. It's you know, a metabolic model in the sense that there are metabolites and things eating them. It's not quite the same as, you know, genome scale metabolic modeling, and that you know, these are fake metabolites. They're not reflecting, you know, actual glycolysis and things like that. So I think, you know, one of the things that I found pretty interesting here is this model naturally lets you ask, you know, here are two communities that we simulate in the model. You know, one has less diversity, one has more diversity. And one of the outputs that we're trying to get from these calculations is almost precisely what Jesse was talking about, right? So essentially, what is the distribution of fitness effects of new mutations? How likely is it, if you knock out a mutation, for it to be beneficial? And I think one of the interesting things to come out is that actually that you still get lots of beneficial mutations, even in saturated communities. And we can kind of understand why that is in the context of this specific model. And then, so that's maybe speaks to what's the potential for evolution happening. And then the other part that I think we find pretty interesting is that a large fraction in this bottom case, like 50% of all successful mutations, don't outcompete their parent. They coexist with them stably instead. And so in this model, you can say exactly, you know, that is actually stable. It's not just a question of you haven't waited long enough for one of them to take over or not. And so, yeah, I am actually really excited about this sort of class of models as helping us get at exactly those kinds of questions in a case where we know everything and we don't have to worry about, you know, mapping artifacts and changing environments and things like that. So I think it's going to be a cool approach.
Okay, thanks. And there was another question for Dr. Shapiro. That one was from Belina. And they ask, does competition support the ecological controls model? And they're imagining adaptive evolution experiments where you can add different strains to the same vessel, and often like one strain will outcompete the others. So I guess it's also in general related to the question where there's particular ecological interactions mechanisms that could drive DBD and EC.
Yeah, so I mean, in the short answer is yes. That ecological controls is sort of associated with this idea of competition for niches, where there's like a set number of niches, and as soon as those get filled, there can only be one member in each niche. So you might have two species competing for a niche, but one will win, right? Whereas in a DBD kind of model, you have one niche, and then maybe two species are competing for it, but then instead of one winning out over the other, they persist, maybe because there's a kind of niche construction where they find a way to split that niche, or one is feeding the other, or one uses another species as a niche. So like the number of niches grows with the number of species. But yeah, briefly, yeah, the kind of ecological controls niche filling model is kind of generally associated with competition, is the short answer. I'm not sure if there was a second part to that question that I didn't answer. Okay. Yeah.
And then the next question is for Dr. Good again. It's from Sander. And they asked if we can look at the speciation within a host as a kind of bifurcation in the sense of a dynamical system. I guess that also alludes to, the effect that you showed, that those like adaptations can sometimes be transient after the antibiotic exposure. So do you think they're fairly dynamic in general?
Yeah, that's a great question. So I think maybe it's worth emphasizing, on the one hand, it looks like speciation within the host because, it's stable. So perturbation could happen. They go up, they come back down. Jay and Tammy found a great example of that in their paper too, where they actually were able to recapitulate it in the lab. So there's some evidence of that actually being true outside of the gut too. But actually speciation, I wouldn't even use that word, because actually most of the examples I showed you, if you look on longer timescales, these are not different species, right? They exchange DNA with each other at sort of the same amounts as like a normal boring species like humans or fruit flies. And so I think it does create this interesting kind of dichotomy on timescale, right? Like you think these are the same species, and yet the genetic differences between them locally in a community can be kind of stable. And it's very weird. That's what I find very interesting about the problem. Most of our sort of modeling toolkits for doing this, it's like once you speciate, that's it. And there's really not a lot of reason to keep sticking together as a full species. And we see this to a certain extent actually in B. vulgatus too, to get to Jesse's example. So what's a little weird about that species is that there are these two really separated clades at the top. In fact, some people call the other one B. dori. It's a whole separate species. And there's actually not a lot of genetic exchange within that. But then if you go even shorter timescales within one of those clades, there's now lots of recombination again, and still lots of diversity. And many of those things manage to coexist with each other in the same host too. It almost doesn't matter, are they sort of these separate species or internal species? And I think that's one of the things I really love about this system.
Okay, thank you. And yeah, we're almost out of time. So let's maybe slide in one last question that was directed at Dr. Shapiro. And the question was that the plateau and the diversity plots look a little bit like they might have also actually a decrease, like or negative slopes there, which, do you think that's a small enough negative slope to kind of, omit here? Or do you think there's something to that, small negative effects that seems to be present in some of those curves?
Yeah, that's right. It does look like that in some cases, where it might not just be a plateau, but actually start to decrease. So that's a good observation. And yeah, I don't really have a good explanation for that, to be honest. Yeah, I mean, maybe that does just get into noise in the data, and I mean some of that might also be I showed some quadratic some quadratic fits, and so like it that's the way that it fits it, compared to in the GAMs. You don't really see it as much. So we did some tests of like what's the best fit, and in general the cubic fits that don't make it go down tend to be a bit better. So it's true, you kind of see. I'm not sure if it's significant, but I don't have a great explanation for why that would be, but have to think about it.
Okay, so thanks so much to both of you, Dr. Shapiro and Dr. Good. It was quite an amazing set of talks and discussion here. And yeah, we'll be going into a short break before we start with our next session. Big virtual applause to our speakers here today, and I hope you stick around a little bit longer. Have a great day, everyone.
Yeah, thanks a lot. Great.
Thanks, Christian.
Yeah. Thanks.